Think about a simple silver charm bracelet. It's a chain with a few unique shapes dangling from it. But a piece of jewelry with this exact architecture is responsible for digesting the food you eat and hunting down viruses in your bloodstream. Inside your body, millions of molecular machines called proteins are performing those tasks right now. Their construction mirrors that of a microscopic charm bracelet. To understand how these tiny machines function, we have to look closely at the standardized chemical links that form their central chain. The diversity of biological life, from spiderwebs to human muscle, rests on this surprisingly simple, repeating chemical foundation. That foundation starts with the amino acid. An amino acid is a small organic molecule that acts as a single link in our biological chain. At the center of every amino acid is a single carbon atom acting as a hub. Attached to this hub are two standard connecting points, an amino group on one side and an acid group on the other. There is a third attachment point on that central hub for a unique chemical tag known as the side chain, or R group. The R group is a specific cluster of atoms that gives each amino acid its own unique chemical personality. Nature uses exactly 20 different versions of these side chains. You can think of them as 20 distinct types of charms that can be snapped onto our bracelet. By combining a uniform connecting backbone with a highly variable side group, biology creates a system capable of almost limitless complexity. When two individual amino acids meet in the cell, they undergo a specific chemical reaction. The amino group of one molecule docks with the acid group of its neighbor. As they connect, they eject a tiny water molecule. This handwritten chemical equation shows exactly what happens. The loss of H2O locks the two molecules together in a rigid handshake known as a peptide bond. A peptide bond is the specific amide linkage that holds two consecutive amino acids together. This diagram illustrates the biological translation process. Here, a cellular machine called a ribosome reads genetic instructions from RNA and rapidly pieces together the correct sequence of amino acids. Inside the ribosome, hundreds of amino acids snap together one after another, forming a long, flexible string called a polypeptide chain. But a linear sequence of linked amino acids is just raw material. To actually perform a biological function, this floppy string must take on a precise three-dimensional physical shape. The newly formed chain doesn't stay floppy for long. The uniform backbone of the chain contains localized magnetic charges. These partial positive and negative charges attract each other, creating hydrogen bonds. This weak electrostatic attraction pulls the chain into two primary structural patterns, a tightly coiled spring called an alpha helix, or a flat, rigid plane known as a beta-pleated sheet. This 3D plot illustrates the physical constraints of these connections. The sharp peak shows that stable hydrogen bonds require highly specific distances and angles to maintain their grip on the structure. These predictable, repeating folds give the floppy chain the essential structural scaffolding it needs to eventually become a working biological machine. Now, we have to look past the uniform backbone and focus on those 20 distinct side chains dangling off the edge. Some side chains carry strong positive or negative electrical charges. Just like standard magnets, opposites attract, snapping together to form strong connections called salt bridges. Other side chains are repelled by water. In the watery environment of a cell, the hydrophobic effect takes over. This effect causes water-fearing chains to hide inside the core, while water-loving chains pull outward toward the surface. This tug-of-war of magnetic attraction and water repulsion forcefully crumples those meat helices and sheets into a highly complex, dense, three-dimensional mass. While the folding process looks chaotic, it is governed by strict physics. The molecule automatically minimizes its total free energy, making this crumpled shape its most perfectly stable physical state. At first glance, the resulting 3D protein looks like a random tangled mess, but every fold and twist happened for a reason. The entire crumpling process works to construct a highly specific, perfectly shaped cavity somewhere on the protein's surface. We call this unique cavity the active site, a localized pocket designed to catch exactly one specific target molecule. Because of its exact shape and magnetic charges, the target molecule docks only when the physical contours and chemical polarities align perfectly, triggering the protein's biological function. Whether a protein digests your food, fights off a virus, or flexes a muscle, its ability to do that job depends entirely on the exact physical shape of this single active site. The complex machinery of biology operates purely on basic chemical attractions and repulsions. The specific linear sequence of those 20 amino acid charms dictates exactly how the backbone twists and exactly how the whole structure ultimately collapses into its final 3D shape. When we understand these precise folding rules, we can trace exactly what goes wrong when a sequence mutates. This allows scientists to diagnose severe diseases that are caused by misfolded proteins. Researchers now use this molecular knowledge to engineer custom medicines, designing synthetic molecules precisely engineered to dock with and modify specific protein shapes. A single chain of amino acids automatically folds itself into a microscopic machine. By mastering the rules that govern those folds, we gain the power to repair, alter, and understand the core mechanics of life.